All right, so it is now 2 o'clock. We'll wait about another minute or so before we start the webinar, just to give people a few more minutes to log on. But thank you for joining today's webinar, and we'll begin in a minute or two. So thanks. All right, recognizing that it's 2:01 and it seems like it's slowed down in terms of attendees joining, I will go ahead and get started with today's webinar, which is Enhancing Research Rigor and Reproducibility. And I'm Yvette Seeger. I'm the Director of Science Policy here at FASEB. And thank you again for joining today's webinar. So here's the agenda for today's webinar. First and foremost, for those of you who are new to FASEB, I will give you a brief overview of what FASEB is and who our members are. And then I will get right to the core of today's topic, which is focusing on our efforts that we've done as a federation pertaining to research rigor and reproducibility. So I will provide an introduction of our involvement in this topic, provide an overview of our 2016 recommendations, highlight 3 new FASEB initiatives pertaining to this topic, and answer any questions that you might have. And speaking of Q&A, you should see a little toolbar on the right side of your screen, and you can type your question into the box and send them at any time. We will be monitoring the questions depending on where they fit. I might answer questions in real time, but we definitely will have time at the end of the webinar to address your questions. All right, so I will go ahead and get started with the background on FASEB. Who are we? So FASEB is a federation of 30 scientific societies, and we represent, through those societies, over 130,000 individual researchers. And our mission is to advance health and welfare by promoting progress and education in biological and biomedical sciences through service to our member societies and collaborative advocacy. And we always get the question of, am I a member of a FASEB society? So this next slide shows you all of our current member societies. You can see that there's a wide range of biological disciplines represented here, which makes this a very exciting organization to work for and with. You know, it really is a great opportunity to provide a broad perspective on topics pertaining to biological and biomedical researchers. All right, so today's topic, research rigor and reproducibility. I will start by providing a little background, our timeline, which is now a couple of years that we've been focusing on this topic from the policy standpoint. But before I get started in the timeline, I do want to make it very clear that when I talk about research rigor and reproducibility, I'm not talking about scientific misconduct, meaning researchers who knowingly are falsifying data or are fudging their results. When we speak about rigor and reproducibility, we are talking about issues in which perhaps an experiment is underpowered. You may not have shared all of the correct information, say about a vector and a plasmid. You aren't doing it intentionally. And I think that is a really important thing that we have stressed every time that we've talked about this topic, because there is a clear difference between knowingly pursuing scientific misconduct and just perhaps your experimental design isn't up to snuff. And we are focusing on the latter perspective today. So how FASEB got involved. So those of you who, a few of you on the line today may remember a time when there were a lot of headlines, both in scientific journals and public-facing news outlets such as The Atlantic, talking about the inability of some biomedical companies to reproduce known published research results. I show a few examples. Those of you who have seen Dr. Larry Tabak from the National Institutes of Health talk about rigor and reproducibility, he has even more headlines. I mean, it varies from scientific journals to daily newspapers, where there were concerns about all of these, particularly in the field of cancer research, the inability to reproduce experiments when by industry when trying to pursue treatments. It became more of a conversation in general press, and why FASEB started tuning into it, not only from the scientific perspective was this a concern, but also as advocates for federal research funding. It's always of concern when there are lots of questions about the funded research being splashed on the front page of the Washington Post. The third image of the PLOS paper on this slide is one in which there was an economic analysis which was a bit contentious, that said, I believe it was $32 billion of the NIH budget was or $32 billion worth of NIH-funded research could not be reproduced, which is a bit startling considering the NIH budget value. But in response, and this has been something that Dr. Collins has done very well as director of NIH, recognizing the signs that this is a concern coming up in the public domain, NIH very swiftly signaled that it would plan to act and address this issue, that this was of concern to the federal agency funding the research. So they made it clear in 2014 that they plan to move forward with some sort of format to enhance research reproducibility. Recognizing that this was something on the radar of the National Institutes of Health, and knowing that we represent 30 member societies and over 130,000 individual scientists, FASEB started thinking of ways that it could engage its membership on this topic. And as Director of Science Policy for FASEB, one of the signature events I get to host every year for our Board of Directors and Science Policy Committee is an annual Science Policy Symposium, which happens usually in May or June. And it was decided in response to all the headlines and knowing that the National Institutes of Health would be issuing new parameters to address research rigor and reproducibility, we opted to make the 2015 symposium on this topic. And the goals of that symposium was to explore the landscape of existing initiatives intended to enhance scientific rigor and research reproducibility. Because we recognized we weren't the first players to the game, that there were people who were already in this field, and we wanted to learn what had already been addressed or what were the specific concerns of the federal agencies, but also what was going on in the private sector, so that we weren't repeating others' work, or anything that we would decide down the road to do would be complementary. And as you can see in the bottom bullet, we were really finding or trying to identify a space for FASEB action in this space, knowing that the National Institutes of Health would be issuing a policy that would affect our member scientists. And shortly after that symposium, our timing was fantastic in this case, NIH released its policy. They released the new policy, which probably those of you who have applied for grants recently, it was released in on June 9th, 2015, went into effect on January 25th, 2016, and laid out some new parameters for any grants or grant applications for NIH funds. So specifically, it looked to add 4 different sections to those grant applications. So effective January 25th, 2016, applications for project funding were required to include an expanded scientific premise. So most investigators already do this. They lay out the premise of their research, but these new guidelines asked for a little more critical thinking regarding the strengths and weaknesses of prior research cited. And this is to make sure that what you're proposing was on the correct path, discussing the rigor of previous experimental design, and attention to incorporation of biological variables and authentication of key resources. So this is looking at sex as a biological variable is a classic example to this part of the scientific premise evaluation. And also, if you're going to use a resource, how are you going to make sure that it is actually what you think it is, or what are your validations of those resources? So that's scientific premise. The second component was expanding rigorous experimental design. And what NIH was looking for here is for applicants to describe how robust, unbiased results will be achieved in the experimental design. That probably seems a bit obvious as you're laying out a grant application, but they wanted a little more detailed discussion about how you were going to power your experiments, what kind of controls. They wanted to leave flexibility in these broad guidelines because every application would incorporate different experimental techniques, but they were looking for the gist of how are you going to make sure that ensure that you were thinking about your experimental approach before you started going down and getting preliminary results. The third component, which I already mentioned as part of the scientific premise, but they wanted to highlight as a separate section, is consideration of sex and other relevant biological variables in your experimental design. So looking for a little more clear factoring in of biological variables such as sex, age, weight, and underlying health conditions into study design, and this can be human and animal subjects. And this is something that came up both on the legislative side, but also on the policy side. If you are only going to use one sex for your animal or human subjects, you need to justify that premise. A lot of times you use what's available to you, or it may seem obvious, but they wanted a clearer description of you're only choosing to use female subjects or male subjects for a specific reason. So that was the crux of the third point. And then the final point was having a better description of authentication of key biological and or chemical resources, specifically highlighting plans for regular authentication of resources to ensure identity and validity. What we honed in on when this policy was released was NIH encourages the research community to inform the development of such guidelines. So this area is looking at the biggest one is authenticating cell lines. Probably many of you watching the webinar use cell lines as part of your experimental design, and there were certainly a lot of reports about you think you're using a certain cell line and all of a sudden they are contaminated with HeLa cells, which completely changes how you would interpret some of your results if they weren't primary cells. So, but on the second bullet, we saw this as a call to, if you're looking at asking the research community to inform development of these guidelines, as an organization that represents 30 professional societies and over 130,000 individual scientists, we thought that FASEB was well suited to at least initiate these discussions and then spin them out to our different member societies. So FASEB, again, being a federation, Regardless of the topic, and in this case, it was rigor and reproducibility, FASEB is recognized as a convener of key stakeholders in policy discussions. And we took this opportunity to say, okay, NIH has put the call out to have the community develop best practices. This is a role that at least FASEB can kind of kick off the conversations, and then perhaps the different scientific communities could take that and make a little more discipline-specific guidelines. And again, reiterating something I said earlier, our goal wasn't to reinvent the wheel. We knew we were kind of the people who show up midway through the party. So we wanted to make sure that we were complementing existing efforts and be very aware of where there was already a lot of work and a lot of work underway, and try to really fill gaps or fill areas that would be unique to the FASEB constituents. We knew that there was increased awareness of the issue and training of early career investigators in cell line authentication. So we purposely stayed away from those areas, but would touch base with those groups as we were working in our own space, because we didn't want to redo what was already going on and actually well underway with those two areas, with training and cell line authentication. But where FASEB was unique or could kind of bump into or overlay or complement existing efforts was the discussion and development of best practices for the use and reporting of mouse models. Since animal research is a core area of FASEB policy interest, we thought mouse models would be a good area where we could find a lot of overlap amongst our societies and antibodies in preclinical research. And then also just engaging other community stakeholders. So we bring 30 societies to the table, but we always want to establish additional partnerships so we could be an even broader convener of the community. And then later on, I will talk about one of our efforts related to the fact that we host a series of science research conferences that we could also use to develop. You're talking about the experimental aspect, but also in the reporting aspect of your research. So those are areas that are unique to FASEB. So going back to 2015, we had our Science Policy Symposium on May 31st. There was a lot of energy around the discussion we had there, and certainly I was given the green light to continue additional activities around this area. We had at that time the policy from NIH. So we pursued a strategy where we held 3 roundtable meetings later in the summer, so in August, September, and October. August, we convened a group to talk about mouse models and what are some of the common pitfalls when utilizing mouse models that we could sort of streamline and say, okay, this is how you minimize variety from lab to lab. September, we did the same thing except with antibodies. And then in October, we brought some of our partners, we partner with them in the DC area, but other professional societies that were talking about rigor and reproducibility with their members to kind of see what they had already done, what worked, what didn't, so that, again, we were being complementary, not repeating existing positive efforts, but also not repeating failed experiments. So, these roundtable meetings had delegates from each of our member societies. We would have 4 to 5 invited speakers, and we had active participation from our member societies as well as NIH leadership. And they were really great dialogues. I've already kind of talked about some of the goals, but, talking about some of the common issues that decrease replicability of research studies, and in our case, we were looking at mouse models and antibodies, developing consensus principles to guide researchers in controlling these effects. And again, we were looking at, early career investigators or those who are just starting their labs, some of the things that you may know further down the line what kind of advice could we give them so that they avoided some of these rigor and reproducibility pitfalls. We wanted to exchange information about other efforts. One of the things, and I'll talk about it certainly later, but it bears repeating here, we don't always, as scientists, talk about our negative results unless we can frame it with some positive results. So we wanted to make sure that we were aware both of what was working in the community, but also things that had been tried and didn't work. And then the last two were really important to us, but certainly the penultimate bullet here is seeking consensus. You're never successful in policy or enacting change if you try to just move forward and say this is the way we're going to do it. There are a lot of aspects to biology, and biology disciplines vary in terms of their needs, but we were trying to find areas that there would be consensus so that they could see a little bit of, oh, this applies to my research in everything that we did, instead of saying, okay, this has a caveat for this discipline. But by having consensus recommendations, it allowed different groups with the more specific disciplines to take those basic guidelines and then really hone them for their specific field. And that is something that we try to do in all of our work here at FASEB. And so our overall goal for these roundtables, we wanted to have the discussion. We knew we wanted to create a resource at the end. We had the NIH policy, so we wanted to prepare investigators to respond to that policy that would go in effect in early 2016. So we ended up we wanted to create a resource to increase awareness and facilitate compliance with the new NIH requirements to address rigor and transparency in the grant applications. And so on the next slide, what we were fortunate to be able to do, we created a series of recommendations that came out of those roundtable discussions, and we released them just in time on January 14th, 2016, to help investigators kind of navigate those new NIH policies, but also provide charges to other parts of the research community. I'm going to pause for a second here on this slide because if you look at your toolbar that you should see on your screen, and you look at the different headers, you'll see a section called Handouts. This report is available to be downloaded as a handout because I'm not going to read the whole report to you because there are pretty dense recommendations. But if you haven't seen this report, it is attached as a handout to this webinar. And to summarize what these recommendations covered, we organized them into 4 sections. We had 3 overarching recommendations that applied to everyone. We were able to boil down our very active topical roundtables into 6 recommendations for mouse and other animal models. We ended up with 3 for research involving antibodies. And then, what I like about our effort, and I might be a little biased because I helped lead it, but we weren't just saying, oh, this is stuff that the scientists have to think about. We were thinking what we could do as professional societies and organizations. What could we do to help enhance rigor and reproducibility? And part of this stems from the fact that most professional societies, certainly those within FASEB, are also publishers of journals where research is communicated. So we had a total of 16 recommendations. I will take the opportunity to highlight our 3 overarching recommendations because these are recommendations that I have, when I've talked about what FASEB has done in this space, I always highlight them because they may seem basic, but until these 3 overarching recommendations are addressed, there will be challenges. The challenges for rigor and reproducibility in the biological sciences will likely continue. So the first one is to use the same terms and definitions when discussing research rigor and transparency to promote uniform understanding of the issue. And if you go into some of the early literature, and even now, people will talk about rigorous research, reproducibility, replicability, And sometimes you mean the same things, or sometimes you're just using these terms interchangeably, but you mean different things. So we highlighted that, and we proposed in the report some recommended definitions, and that's when we start talking about those terms, that's what we prefer to use. But it seems like a simple thing, but if you listen to talk about rigor reproducibility, a lot of times people will use these terms interchangeably. So that was our first recommendation. The second was the need for stakeholder dialogue to develop uniform expectations for describing agents, laboratory tools, and protocols in scientific communications. Again, this seems very obvious, but even though we continue to talk about it, even before this controversy was coming through the general press, you see different formats. It can either be driven by the journal or it's just by the different disciplines where they will have different ways of describing antibodies. So the antibody section of the report is actually quite fascinating because you will see a lot of variance even in scientific publications as to the type of information that is reported about an antibody used. And that's what we're getting at, is that we should all use the same language. Just like for biochemists, there's the agreement that if you have an enzyme, it will end with ASE, so that it's an ACE. And so trying to get that same language or that same sort of feel for how you're describing reagents of a certain type was what we were getting at with that second recommendation. And then the third one was to reinforce good laboratory practices as part of research training, making sure that your graduate students, your postdocs, anyone that's a trainee, learn key skills in record keeping and reporting, use of standard nomenclature. This came up a lot, both with antibodies and mouse models, so I can only imagine that it dips into other areas of biology as well. Experimental design, and proper use of statistics. And this latter point is something that has been highlighted a lot by NIH's efforts. Are you powering your experiments properly? The experimental design and capabilities have changed so much in recent years that, do you need to take a step back and make sure that you are using the proper statistics for the experiment? Are you using the proper tool to measure what you're trying to measure? So those were our three overarching recommendations. And, I mentioned the four recommendations that were geared towards professional societies. One of them was pointed back at us. Again, looking at what could FASEB do. And the last recommendation is the role of FASEB. And FASEB will continue to facilitate the dialogue on enhancing rigor and reproducibility by synthesizing the views of the research community regarding clarity of the NIH policy, challenges encountered, and resources that could help streamline compliance. And this is something that we have continued to do since we embarked on this effort in 2015, as the guidelines were put in place in 2016. Certainly in 2016, we heard a lot of feedback about how do I write this? How do I write this in my 12-page proposal? From the reviewer side, I'm not quite sure how to review this. What are the guidelines for peer review? And so we've continued to relay those concerns back to NIH to help them make the policy better or provide better instructions so that you aren't just missing the mark because you didn't understand the policy, which is always a big concern for us here at FASEB. So that is our 2016 report. Do we have any questions? Are we good to roll? Because if there aren't any questions, all right, so hopefully the person who said they can't hear any audio can hear it now. Yes, okay, and this is a great time to answer this question. Will the webinar be available on the FASEB website? Yes, there will be a recording of this webinar on the FASEB website, usually within 24 to 48 hours of the webinar. So if you would like to share it with your colleagues, please do. And I do believe we will share the slides separately, but my contact information will be at the end if you're interested in receiving the deck. So now that we've cleared the questions, I always like to make sure I address them. So we did our report in 2016. Now I'm excited to talk to you a little bit about our follow-on activities since we released those recommendations. And the first one is our Scientific Research Conferences Transparency and Rigor Initiative. So giving you a little background on the timeline. We were approached by a colleague at the National Institutes of Health, where we were chatting about what are effective ways to provide more transparent communication of research methods and findings in short presentations and posters. So a lot of the concerns that have been raised in the initial discussions of this issue in the general press were concerns about something that had been published in a journal. Somebody went and tried to repeat it later. And that's what kicked off the conversation. But where do investigators start to have a conversation about work? Where are they starting to get critical feedback? And that's usually at scientific conferences. Anyone who's ever given a 10-minute talk knows that you are pressed for time in terms of what you can say and what you can show on the slide, and what also, depending on where you are in terms of your research, what you're willing to show. And so we wanted to think of the fact that so many scientists every year go to conferences, be it a FASEB Society conference or our science research conferences. This is where they're exchanging a lot of information. How could we improve the correctness of that information, or at least manage expectations of what is being shown on the screen or talked about at a poster? So in February of 2017, we went to a workshop that was coordinated by the National Institute of Neurological Disorders and Stroke, or NINDS, and several conference organizers of various size, so very large meetings, very small meetings, and then middle of the road. And we discussed the feasibility of integrating icons into conference presentations so that you could have a little cheat sheet as to if you couldn't always, if it's a poster and somebody wasn't there to describe it, or, if you were looking at a slide very quickly in a short talk, you would kind of get a gist of some of the variables and the controls that were applied. And after that conference, FASEB, through its scientific research conferences, decided that they were willing to embark on a pilot. This is the great part of FASEB having such a multifaceted business plan with a conferences group and also a journal, is that we can conduct some experiments of our own. And so we initiated a pilot in 2017 for the conferences last summer. It was voluntary participation, and we tested the use of 4 icons to communicate specific aspects of experimental design. And then following the conference, there was a survey that was distributed to everyone who attended the conference, and there were several questions pertaining to rigor and reproducibility and the transparency and rigor pilot in there. So, we were looking for, at that time, the NIH policy was a year in. So were scientists aware of NIH's rigor and reproducibility initiative, and also get their feedback on some of the icons tested in the pilot. So this slide summarizes some of the findings from that first pilot. As you would expect, there was still general skepticism about the broader NIH rigor and reproducibility initiative, and what came out of those comments was a lack of clarity as to what was expected that I highlighted earlier in this talk. It was NIH left it intentionally vague so that there was room for interpretation for different fields of science, but that was still generating some skepticism. Pertaining to the icon pilot, some people are aware, others were not, so we needed to do a little bit better awareness and socialization of the pilot. Apparently, we highlighted the icons, but we didn't provide any specific guidelines regarding size or uniformity even of the icons. So I'm picturing individuals posting slides with very large smiley faces, which is why I giggle every time I think about it. But we really did. If we were going to do this pilot, we should have some guidelines about the size and the placement of these icons. Then there was also a request to expand the icons to be a little more relevant to different fields of research. The first round were very focused on animal research, which not every conference attendees found applicable to their work. So we made some updates for a pilot that is underway right now in the summer of 2018. So addressing some of the feedback from the survey, we improved the socialization of the pilot. There is actually a web page, and one of the last slides of this presentation has all the hyperlinks, so you can quickly find this stuff. But a more detailed description of the pilot, and that this could be included in the meeting materials, so that people weren't surprised by these random icons that were attached to slides. And then incorporating different aspects for the meetings, so that the organizer, when introducing or opening the conference, would say, we're also allowing voluntary participation in this pilot. So if you see these icons on slides, this is what they mean. Isn't that cool? Or, do you have some negative feedback that you would like to share? We also spent a lot of time enhancing the instructions for icon use. Simple things, including the icon next to the image where it was relevant, not just on the title slide. Again, we learned a lot from the first pilot. We provided uniform downloadable image files so that everyone was using the same image. We also offered specific guidance regarding appropriate image size. And so this slide shows you the 8 icons that are in use for the 2018 pilot. And we, again, tried to have some wide variety so that pretty much any discipline could incorporate an icon if they wanted. Some of them, I've gotten some feedback whenever I've presented this, like preliminary or confirmatory, or, whether or not it was blinded. There's varying reaction to the use of the icons, and we expect that. But it's been a pleasure in the opportunities where I've presented this as an opening session, and then I've watched presentations of the science later, and I've watched scientists actually add preliminary or confirmatory to their slides, because it saves them having to say it. And it was great to see them kind of naturally take on the idea and test it for themselves, because they were thinking, oh, okay, what am I presenting? And it helps when a slide is labeled preliminary. Okay, so this has been done maybe once, maybe twice. There are certainly going to be additional experiments, and they found it. The feedback when they've used it that way has been very positive. So that is our SRC Icon pilot. Another effort that we hosted last year is a communication workshop. And this was a partnership with the National Institute of General Medical Sciences, because we have partnered with them quite a bit on rigor and reproducibility. And they were an active participant in our roundtables in 2015. And one of the things that came out of the shared interest in improving rigor and reproducibility was to come up with ways, more effective ways to convey the importance of basic biomedical research, but without the need to hype it. And I think all of you can think of a headline or several headlines. Coffee is great for you. Coffee will cure everything. Coffee is bad for you. You shouldn't do this. Red wine is great. No, white wine is great. And there is this, how do we make sure that research findings from a single study aren't hyped, was the main emphasis of this workshop. And the goal of this workshop was to identify best practices for communicating basic biomedical science across the range of potential stakeholders. So this workshop was held last June, was called Responsible Communication of Basic Biomedical Research, Enhancing Awareness and Avoiding Hype. And we centered the agenda around 4 key questions. The first, how does science communication affect the biomedical research landscape? Second, how does science communication affect public perception of science? Third, what are the goals and incentives of science communication? And fourth, how can all stakeholders work together to improve communication of scientific results? And we were super fortunate to have a great group of people who were willing to come to Bethesda in June to talk about this. We did 4 different panels where we mixed and matched speakers from different backgrounds. But what I loved about this, in addition to having Erica Check-Hayden as our keynote speaker, this wasn't just a bunch of scientists talking to each other. We had biomedical researchers, we had researchers from the field of communications, we had a representative from industry, we had several journalists, we had marketers, and we had the government and policy perspective. And so we would balance and we would rotate the speakers to the panelists' seats at different times, but they were all engaged in the conversation, which really gave us a 360 view on this topic. And so we identified several challenges, some of which you could probably easily cherry pick, but one of the key issues is always the public understanding of science, and I think we can see this in our day-to-day. You turn on the TV, science is complex. There are a lot of uncertainties. Science doesn't happen quickly, especially for some basic biology studies. It is a pretty lengthy process to get from that first experiment to something that might directly affect a patient. As scientists, there is a pressure to connect results with clinical applications, and that is a challenge for basic science. That is NIH's challenge every time it asks for additional federal support. How do you connect this very basic finding to human health? There are varying incentive structures in different organizations. A scientist will want to get their work published in a scientific journal because that is how they advance their career. But universities want to show the community that the research going on at that university is benefiting the community, creating jobs. So there are all these different tensions about the different audiences of this research. There's also the challenge of the uncertainty associated with when and how to communicate findings. Do you just take one study, or do you bin a group of research studies and then release them? I think all of you could probably think of the instance where there's one study that's really interesting, but it's not going to be the magic bullet. So there were several recommendations that we came up with. The first one deals with effective communications and providing communications training and resources to scientists throughout their careers, not just waiting until they have their own lab. I think it's very important for trainees, meaning grad students and postdocs, to learn how to communicate effectively with a broad range of audiences. Telling a story. This storytelling, this is what scientists do, and they do very well with other scientists, but sometimes you might have to change the story a little bit to match a public audience. Using metaphor, imagery, and illustrations to explain complex concepts or techniques. You may have to simplify the message to reach different audiences. And this is a really exciting time to communicate your science because there are so many different platforms. And they can be visual, they can be audio, and you should use all of those platforms to communicate your science. The second bucket of recommendations pertain to incentives for communication, emphasizing the process. This is something that we talked about quite a bit during this workshop, reminding the public that it's not just one experiment equals cure, that is a whole scientific process, that there's a pipeline to take a very basic research finding to make it into a pill or a treatment. Developing and following a logic model to outline both long-term and short-term communication goals and establish appropriate tactics. Sometimes you'll use a certain research story in one way that you wouldn't use later on. So you need to figure out your short and long-term communication goals so that you can maximize the use of the research findings. And then the last part of this bucket of communications or bucket of incentives is considering which news outlets can best reach the intended audience. Not everything, and I think those of you who have published scientific papers, not everything goes into the big 3 journals, and you need to find your appropriate audience for your field. And that is the same thing even with dealing with public-facing outlets. The last slide about these recommendations is reconsidering press releases. This was an amazing part of the workshop, and then the next slide will show you where everything is archived. You can watch this. The discussion around press releases was fascinating. Not everything is worthy of a press release, and in fact, our scientific journalists said that they really don't follow press releases. They would rather talk to the scientists. They would rather go to a conference and listen to how the scientist interacts with the audience about their research as opposed to following a lead from a press release. And one thing that definitely piqued FASEB's interest was identifying scientists who can comment on stories in the news, not the individual who did the research, but experts, because the more you can get feedback from others in the scientific community commenting on a scientific story, it helps build trust with the public. And the take-home message from the whole workshop was embracing and helping others understand the uncertainty of scientific inquiries, and that is the biggest challenge for scientists. As I mentioned, all the meeting materials are on the FASEB website. There is, when you go to Science Policy Advocacy and Communications, there is a tab Communicating Science, and there is the Communicating Science workshop. And so all the materials there, including an executive summary and a full meeting summary, are there. But you can watch the 4 different panels, and it was a great day. So our last thing that we've done, and luckily this is very brief because I'm recognizing that I want to leave time for Q&A at the end, is FASEB BioAdvances. So FASEB BioAdvances is a new international peer-reviewed open access journal that is a partnership between FASEB and Wiley. And it is in addition to our existing FASEB journal. And in addition to reports of new findings, the journal considers articles with confirmatory results. And those of you who subscribe to FASEB mailings may have gotten an email last week that talked about how FASEB BioAdvances will publish articles as research reproducibility reports. So these are articles that successfully or unsuccessfully replicate methods and results of previously published articles. This is something that we identified back in 2015 in our 2016 report, that there really, journals need to make room for confirmatory analyses or replicability analyses, and we're really excited that our new journal has decided to pursue this option for investigators so that as you're conducting research and trying to confirm results, or you find that you can't, there is a place for you to submit that so that it can go in the scientific record. And hopefully, we talk a lot about science being self-correcting. It can only be self-correcting if we're effectively communicating where we've had challenges reproducing existing results. So I wanted to be sure to highlight that new initiative coming out of FASEB through the FASEB BioAdvances journal. As I mentioned, I will be able to make these slides available to you because there are these hyperlinks to all these initiatives that I highlighted. If you wanted the report, you are able to grab it as a handout from your right-hand toolbar. But these are all your hyperlinks to reach the SRC Transparency and Rigor Initiative, our NIGMS Communications Workshop, and the link to FASEB BioAdvances if you want to learn more about the journal or the types of articles it accepts. So with that, I will turn it back to you if there are questions, and Jocelyn is going to open that for me so that I can see. I see a question. Fantastic. All right. So I think I addressed the first question from Alice, which is negative results. report experiments that were tried but did not work. How will you implement that? We're hoping, and FASEB BioAdvances is one of a couple of journals that is publishing, the replicability experiments, but journals are also realizing that you don't always, they should be more accepting to publishing experiments that don't necessarily have a clear outcome. I call it the Journal of Negative Results. We always joked about that when I was in grad school. And there is a push from scientists as well as the journals to say, hey, if something didn't work, we should keep track of that as well, particularly in research that you're hoping will go into humans. I think it is important for people to know that there isn't necessarily a positive impact. And so, the more scientists can push for journals with which they're involved to publish those types of findings, that would be helpful because there's a clear need for it. What are icons? So hopefully my slides help this. Icons are the other word that we use to describe this project, and I will only say it once. I won't even use that term, but they're basically pictures to represent a phrase or word. So if you go back to, we won't go back to the slide, but that there was a check mark for confirmatory analysis. There was a stamp for preliminary. And so that tells you, when you see that stamp of preliminary, okay, this is a preliminary result. Or you see the check box, oh, that's a confirmatory study that they've done this several times. And another question, the areas and criteria identified for basic research are a great start. What are the possible steps to pursue similar recommendations and guidelines for clinical studies? Oh, fantastic question, Michael. We're hoping that, and what I've highlighted here are just things that we've done as a federation. At the beginning of the presentation, I showed you our 30 member societies. I hope at some time we can have future webinars. Several of our societies have taken on their own rigor and reproducibility initiatives and projects, and really checking in with your scientific society or a coalition that you're part of. Really, you just have to convene the right stakeholders, because that is the next step, talking about what is needed for clinical research. Those conversations need to happen, and finding, I think what would be particularly helpful is finding the scientists who bridge the basic to clinical, the translational gap. I think that is a great opportunity. We do have some member societies who have a little more of a clinical focus, and my goal, kind of separating our efforts, is we always kind of take a step back every year and reevaluate what's needed. You've seen 3 follow-on projects. This might be something that a potential partnership down the road. We didn't necessarily initiate it, but finding the right stakeholders and convening them, and finding, I would suggest, 2 to 3 topics that you wanted to make recommendations on in the clinical space, I think would be a great start. All right. The next question, regarding the reduction of hype, the workshop described seemed to assume that scientists speak directly to media. However, many of the media reports we would describe as including hype are generated directly as press releases by university communication offices with little or no consultation with the affected scientists. Whether hype is generated by non-researchers or by the researchers themselves, how does FASEB propose to reduce this practice? Great question. That was certainly a topic that came up. And one of our outputs back to our scientists following this workshop was, and we talk about it a lot just when we're referring to academia in general, is making sure you know the different offices. It's not just, and the onus just isn't on the science, and it's not just on the communications or marketing office. Studying that was that recommendation regarding the logic model. Having the scientists interact with those offices, knowing what steps need to be taken, because somehow that science got from the lab to the press office to write that press release. And we talked a lot about in that workshop, how do we make sure that the scientist is one of the people that they speak to? And so that requires a lot of discussion about within the university or the research organization to make sure that all the right parties are talking to each other. And really, it came down to the bottom line. It comes from the top that there should be that process, and it should be across all different fields of science. But yeah, it's not something that happens overnight. But making sure that there's this awareness of the different parties that are involved in those types of communications is really important. Could we see the list of icons again? Sure. I think Jocelyn can back up the slides, and then I will go through. But that is also available if you go to the FASEB Science Research Conferences website. They're there, but we will back up so that you can see them. The next question is, how would you go about taking someone's research and using the same idea but with a different population? I think that's probably, I think that's part of the scientific process. Generally, you would have to consider how close your existing, and I don't want to get too much into experimental design, but in terms of rigor and reproducibility, you would have to look at the variables that would be similar in the two different populations and what would be different, and thus what would you have to describe. I could take that question offline if you need a little bit more, because that gets the way I'm reading it gets a little bit too much into design, and I don't want to talk about too much into experimental design. Oh, great question, as I get to read them before I say them. Is FASEB or anybody creating a clearinghouse where investigators can share their experiences with, say, antibodies or cell lines? I spent thousands of dollars on bad and effective antibodies, join the club, and probably others did too. Yes, there are some warehouses, so there are generally about resources to help investigators address concerns about rigor and reproducibility or learn about experimental design. I will give a shout out to the National Institutes of Health and NIGMS, National Institute of General Medical Sciences. They've really collated a bunch of resources together. We were going to duplicate those on the FASEB website, but we're still trying to figure out if that is the right way to go, because they keep updating their website all the time. There are some repositories, and actually we had a representative from one where you could we called it the Yelp of antibodies. They do exist. However, a lot of them, and I haven't checked, I'll be honest, I haven't checked in the past couple months, but they tend to have difficulty maintaining support. So, they'll start the Yelp of antibodies and then they kind of fizzle out. So again, it's kind of those of you who were conducting science in the early days of the Human Genome Project and all the online databases, it's only good information if it's maintained. And that's certainly, there are starts of it, but I think there does need to be a broader community discussion about how would we maintain one of those resources that is very important to talk about or to make sure that investigators are making the right choice. That said, also during our antibodies meeting, we had an industry representative who wanted feedback about the type of information scientists really wanted to see on the product spec sheet, and that was to be part of that discussion was pretty amazing, because in a good way, because they were really taking that feedback and saying, okay, what we provide is actually not what you need. It's interesting to us, but it's not helpful to you. And so I'm hoping that perhaps that could be a way where you could at least get better information from the vendor about the appropriateness of a particular antibody for a specific experiment. That was a huge thing that was highlighted, is that sometimes you buy an antibody thinking, oh, this is perfect for my Westerns, but it's actually optimized for a different technique. So, all right. Is there any effort to promote the inclusion of information of animal diets in publication? Yes, great question. Yes, talking about animal diet, talking about all the different variables, talking about housing when you're talking about animal research. There was a lot of discussion about how much space you have to describe these things, but how important it is when you're trying to replicate these experiments, talking about the chow, talking about the housing, even talking about when you conduct the experiment. There is a push, and if you look at our report and recommendations pertaining to animal research, you'll see that there are parts that deal with this specifically. Great question, thank you. What expectations of good laboratory practices would be for small-scale discovery environment? I think that is beyond what I could cover here, but I will try to follow up with you later. Okay, thank you for this feedback on the icons. Is there any overlap of these efforts with NIST? Great, the National Institute of Science and Technology and Standards. We speak in alphabet soup here in the Beltway, so sometimes it's like I know what NIST is, but yes. Now FASEB isn't specifically partnering with NIST, but the National Academies is conducting its own review at congressional request of how can we, when you, as I mentioned at the beginning, when you have scientific issues that make common headlines, congressional leaders will read those headlines, and so they actually charge the national academies to conduct a study of how could they enhance rigor and reproducibility of all science, not just biology. NIST did present some of its efforts in conjunction with a panel that I talked about what FASEB did. And so it certainly has opened the lines of communication, and certainly NIH and other federal agencies are talking with researchers at NIST about what they're doing, and that is a big thing, is having that open communication. So they certainly are on the radar. All right, yes, we will make the slides available, and actually what Jocelyn can do is go to the last slide with my email address if you want to follow up with me. In terms of, there's a question about what sort of statistical training should be incorporated into training. Well, first of all, the simple answer to that is, I have a PhD in genetics, and I did not take a stats class as part of my training. Yet a lot of the work that I did, luckily I'd done it as an undergrad, but are you making sure that your trainees are aware of different statistic models, appropriateness of different models to different experiment types? That has been identified as a clear gap in research training, and a lot of times it's kind of you're passing the information along through the PI or the postdoc Sherpa who says, this is how I've done it, these are the models I've used, and so giving our trainees the confidence to explore different statistical models or more appropriate models for the type of experiments they're doing or the technology they're using should be there. And again, that might be a little field specific, but I would say just even the biggest gap is the lack of statistical training. Let's see. Our society annual meeting starting to encourage a standard set of icons on presentations for posters or platform slides. So we have the standard set for the FASEB SRCs. We're hoping that as people start with those 8, that they'll think of different icons specific for their field, and that's what we try to get out of the survey every year is what is common for your field. If you have suggestions, we're always happy to add them to our bank, but we hope that as the idea moves forward, there was a request for information from the National Institute of Health regarding icons that could be common, and I don't know the status of their analysis of those comments at this time, but we're hoping to get there. We're not quite there. And has FASEB provided specific guidelines for validation tests of antibodies? We offer a framework in our reports, but again, we tried to leave room for the actual disciplines to take the lead there. We wanted to provide them a framework of some of the things they should consider. So we did not take a particular stand on, these are all the boxes you should check, but these are some of the boxes that that we think would be appropriate for you to consider. Because at this time, we want to be flexible, because nothing is worse than implementing a rule that you find out down the road just isn't working, because then once people try it and it doesn't work for them, they will never come back. So, we're walking a fine line in terms of providing a framework but not being too directive. So hopefully that helped. But again, here's my contact information. If you would like to email me, or would like a copy of the slide deck, I'm happy to share. But I really want to thank you all for tuning in, and those of you who have stayed an additional 5 minutes. Seeing no additional questions, I will go ahead and end the webinar. And thank you once again for your participation, and do look for the recording in the next 24 to 48 hours. Thank you so much.